So the ribosome is a cellular machine which is made up of a lot of different proteins, almost 80 in number, and 3 different RNAs. So it's a very complicated structure. And its role in the cell is to physically knit amino acids together to make proteins. And defects in this process have been implicated and increased as increasing cancer risk in a lot of different tissues, but very often in blood. And so while this was known for quite a long time, maybe a decade or more, no one really has any understanding as to how this risk is, why the risk is increased. And so that's why our observation is really an important finding, because it's the first insights into how a ribosomal protein mutation can increase risk. And it does so by increasing the expression of a gene called Lin28B. And this is a gene that's highly expressed in stem cells. And when this gene is overexpressed, as occurs when RPL22 is mutated in any kind of cell, it makes that cell behave more like a stem cell. And this is in fact what increases its cancer risk. Because Lin28B actually is involved normally in enabling oncogenes to be increased in expression, and that's really how RPL22 links to risk, by increasing Lin28, which then increases the expression of oncogenes.
Okay. For this study.
Okay, good question. The simple reason is that Fox Chase was founded on the principle that we must first understand normal biology before we can understand how that biology is perturbed in cancer. And so, sort of an extension of that way of thinking is that any gene that we find that's essential for a normal biological process, we think is very likely to be involved in transformation. And RPL22, we demonstrated to be required for the development of a type of blood cell called a T lymphocyte. And so because RPL22 is required for the development of those cells, we thought it might also play a role in their transformation. And so that's why we focused on L22 as a potential tumor suppressor, and that's why we focused on T lymphocytes as a potential target cell population where that transformation might be realized.
Okay.
So in a few ways. One is that the identification of RPL22 as a tumor suppressor enables us to use that information to predict how disease course will go. We found that the mutation in RPL22 occurred in patients with a leukemia called T acute lymphoblastic leukemia, and it occurred only in those patients with the worst prognosis. So by looking for mutations in this protein, it will alert clinicians that these will be difficult cases to handle. And so those patients must be monitored more frequently, and also probably treated in a different way. And so sort of the second part of that is that we hope to be able to understand how RPL22 talks to different pathways in the cell so that we can devise new strategies to treat those cancers that may be less toxic and more